Afternoon, everyone, or good morning for my case in the United States. I would like to thank everyone and the organizers for the opportunity to present my research with you today. My project that I'm going to be presenting today is sex-specific colonic mitochondrial dysfunction in the indomethacin-induced rat model of inflammatory bowel disease. So, as most of you know, inflammatory bowel disease is a term for two conditions, Crohn's disease and ulcerative colitis. Both of these conditions are characterized by chronic inflammation of the GI tract, and this prolonged inflammation will result in damage to the gastrointestinal tract. In the United States alone, around 3 million people suffer from inflammatory bowel disease. If you look at this picture on the right, the high incidence of inflammatory bowel disease is mostly in our westernized countries, the United States, Canada, and Europe. There are some incidents or moderate incidents in Australia and Southern Africa. However, there are a lot of areas where it's unknown. So either people aren't seeking medical treatment for their GI issues, or it's just not being reported. IBD is more prevalent in women than men, so both the incidence and severity of Crohn's disease is increased in women compared to men, and the gender of female is going to increase your risk for a relapse of ulcerative colitis. So as you can see here in this study, there's an estimated increase in women compared to men. Another interesting fact is that women have a lot of changes in their hormones throughout their life, which is going to result in changes due to the disease. So in this graph, for example, the odds of developing Crohn's disease is actually higher in males in childhood, but once we hit puberty, women tend to have a higher increase in odds of developing Crohn's disease, and this will continue to fluctuate throughout their life due to pregnancy, menstruation, and menopause. So what are the causes of GI inflammation? Well, there are a lot of key players that are involved in maintaining a healthy gut. So first off, we'll start discussing the bacteria. You have your good bacteria, they're going to produce the toxins that will kill the bad bacteria or attack the bad bacteria. They also produce substrates that are utilized by the epithelial cells to generate ATP. When the good bacteria come in contact with those epithelial cells, they're going to cause them to generate some ROS that's used for signaling purposes in order to induce proliferation of more epithelial cells to generate more ATP. You also have that decent sized mucosal layer that's going to also prevent any leakiness from occurring. However, when your gut is unhealthy, those good bacteria are few and far in between. Your bad bacteria are producing a lot of their toxins, and they're generating and they're growing. Without those good bacteria, we don't have those substrates that can be utilized by the epithelial cells. We're not creating as much ATP. And one of the reasons why those good bacteria are few and far between is because our mitochondria are damaged and producing a lot of ROS that is going to kill those good bacteria. Therefore, that ultimately is going to lead to that mucosal layer being thinner, and it's going to cause leaky gut. And these PAMPs will then transfer through, inducing inflammation. So the goal of this study is to explore the role that mitochondrial dysfunction plays in the development of IBD. The reason why I chose this project is these are some biopsies from patients with ulcerative colitis, and you can see that there are some mitochondrial changes, though the studies don't quite match. So what I wanted to do is explore this in rats in order to clarify potentially what's going on and have it more cumulative mitochondrial study. So my overall hypothesis for this study is that inflammatory bowel disease will increase mitochondrial dysfunction, mitochondrial ROS, inflammation, and the severity of the disease symptoms will further increase. So there are several animal models that can be utilized to study inflammatory bowel disease. There are the chemically induced models and there are the transgenic or knockout models. Most of these are in mice. So since I need to use rats for my study to increase the mitochondrial yield and mitochondrial amount, I'm going to focus on the chemically induced models. And for this specific study, we're looking at indomethacin, which is more of a Crohn's disease model to work with. For example, all it requires is 2 subcutaneous injections that are 24 hours apart. So we used 8-week-old Sprague Dolly rats to start our study with. And again, exactly 24 hours apart. Rats are monitored and sacrificed at day 2, which is our peak of the disease, which I'll show you in a minute. We collect the colons and do histology and tissue homogenates for mitochondrial studies. So just some markers of the disease. At day 2 and 3 is typically the peak of the disease in this model. It's an acute model, so eventually the animals will recover. So it's like a flare in a way of Crohn's disease. At the peak of the disease, they're going to lose weight. They're going to have lower food intake because they are sick. The females actually lose more weight than the males. So the females are getting slightly sicker than the males, but not too much. Also, another marker for the disease working is a decrease in colon length and an increase in colon weight. So we can see in both males and females, we have a decrease in colon length. However, colon weight, either by body weight or by colon length, was only significantly increased in the females. When we looked at histology, we see both males and females show alterations in their size, shape, and number of their Peyer's patches, and this has been associated with IBD. So this is just a representative image, but we have sections throughout the colon to examine this. So the next thing that we like to do is to look at mitochondrial content in our samples. So we can do that by cardiolipin, which is a phospholipid in the mitochondrial membrane, or by citrate synthase, which I'm going to show in a minute. Both of these mitochondrial markers show that there's a decrease in mitochondrial content in our samples in the males with IBD, but no change in the females. So again, with citrate synthase, we see a decrease in the males, but no change in the females. So we'll use citrate synthase throughout the rest of my presentation to normalize to mitochondrial content because this is an easier assay to do than the cardiolipin. Next, we want to measure mitochondrial respiration. We will utilize the Ouroboros fluororespirometer that will, in the case of this study, measure mitochondrial respiration simultaneously with ROS production using Amplex Red. So we have 4 different substrates that we use. The first is glutamate and malate, which we'll term complex 1 driven respiration, which provides electrons via NADH to complex 1, which then will transfer those electrons to 3 to 4 and ultimately to oxygen to create water. The next is succinate, which is complex 2 driven respiration, which will provide electrons to complex 2, 3, 4, and ultimately to oxygen to create water. Long-chain fatty acid oxidation will use oleate. Long-chain fatty acids require the additional acylcarnitine transporters or CPTs to enter the mitochondria and go through beta oxidation. Medium-chain fatty acids, or in this case, octanoates, do not require transport into the mitochondria, so they can just freely cross the membrane. Once these fatty acids enter beta oxidation, they will produce substrates that will be utilized by complex 1 or complex 2 in oxidative phosphorylation to consume oxygen. So what we found was with complex 1 driven respiration and complex 2 driven respiration is that the females have a significant decrease in respiration compared to controls. So IBD is causing a decrease in respiration. The females, when compared to male controls, have a higher respiration rate as well. Again, with fatty acid oxidation, we see that the females have a decrease in fatty acid oxidation in IBD. Males have no change. The females have a slightly higher respiration rate for medium chain fatty acids compared to males. When we look at respiratory control ratio, which is a marker of how well intact your mitochondria are or how well they can generate ATP. So we usually take the rate with ADP present. So this will be running full oxidative phosphorylation. State 4, which is here, or state 2, which is here, will be leaky without ADP present. This is just how leaky those mitochondria are. So if we do this divided by state 2 or state 4, in this case we use state 2, you'll be able to determine how well intact your mitochondria are and how well they could generate ATP. And what we found is that the females have a decreased RCR, meaning that they're not as intact and they're not producing as much ATP as controls. The males have a slight decrease, but it's not significant. Right. So what we found, again, using our Ouroboros, we saw that there is a increase in ROS production regardless of substrate in both males and females. There's a trend for the males to have a slightly higher ROS production in just controls compared to females, but that is only significant when we're looking at medium chain fatty acid. Now this is percent electron leak, which means the amount of electrons that escape the electron transport chain and were converted to superoxide instead of going to oxygen to create water. Then we wanted to look at each electron transport chain enzyme in oxidative phosphorylation and see how they were affected. What we found is females are the only ones that showed a decrease in the disease state, and that is in complex 1 and complex 4. Other interesting things that we found was that the females have a higher complex 2 and complex 3 control activity than the males. There is a trend for there to be a decrease in the male controls compared to the female controls. However, that is not significant. So we wanted to explore next how the males have an increase in ROS, but no alterations to any of their electron transport complexes. So we sought out to look at these antioxidant enzymes. So first, there was no changes in superoxide dismutase, no changes in glutathione peroxidase, no changes in glutathione reductase. However, we saw that the males had a decrease in catalase activity. And please remember this for a future slide. So we think that targeting mitochondria may prove to be an optional treatment, especially in this model. So we chose to use mitotempo. Mitotempo is a superoxide dismutase mimetic that will be accumulated in the mitochondria and it will scavenge for superoxide. So it's entering those pathways here. So first we wanted to see if mitotempo is improving our disease at all. So if you score the disease activity score, that is usually by body weight loss, stool consistency, inflammation, and presence of blood in the stools. If you look at the males, they have a disease activity score usually around 6. The males given mitotempo after the 2 injections of indomethacin didn't show a change. The prophylaxis treatment, which means we gave mitotempo before we even gave them indomethacin, we can see a slight decrease, but it's not significant. In the females, however, they have a slightly higher disease activity score than the males, but it's not significant. And when we give them mitotempo, either the treatment or the prophylaxis, we see a decrease in that activity score. So next we wanted to see if mitotempo is going to improve the level of reactive oxygen species produced in our IBD animals. So on the left here we have females, on the right we have males. As you can see, mitotempo, regardless if we gave it as a treatment after indomethacin or before indomethacin treatment, you see that it was able to decrease the reactive oxygen species, but in the males, it was not. So the males have a decrease in catalase activity, and mitotempo is a superoxide dismutase mimetic, which is going to increase the level of hydrogen peroxide, which is what is measured by our Amplex Red assay, which might be why we are seeing an increase in the males or no change in the males, because we're not able to consume that hydrogen peroxide by catalase. Additionally, when we looked at the females, those individual complex activities that were affected by the disease, which is 1 and 4, we saw that mitotempo actually improves the activity of those enzymes. Then when we just looked at inflammatory cytokines to see what was happening in our model with IBD, again, I'm only showing you females here because they were the only ones that responded to mitotempo treatment. You see that there's an increase in C-reactive protein, an increase in TNF-alpha, and an increase in IL-1 beta. When you give them mitotempo, all of these inflammatory cytokines decrease. So just the overall conclusion for the mitotempo treatment, it seems to be a great treatment potentially for females. However, for the males, we need to potentially design a catalase mimetic in order to reduce that hydrogen peroxide level in the males, though this can be difficult. Okay, so overall conclusions were that we showed that the female SD rats have an increased disease severity compared to the males. They lost more weight, they had more inflammation, they had more issues. We also demonstrated that there are differences in mitochondrial function between males and females, which could contribute to their response to indomethacin in this disease. We also showed that both males and females have an increase in ROS, but they have a difference in how their mitochondria are potentially producing ROS. For example, the females have a decrease in complex 1 and complex 4 activity, which could lead to a potential for those electrons in the electron transport chain to escape to generate ROS. But the males have a decrease in catalase activity, which also would result in an increase in hydrogen peroxide and further oxidative damage. Targeting mitochondria with a superoxide mimetic like mitotempo proves to be a potential option for the females using this model. However, for the males, it's not a viable option, and making one of those catalase mimetics would be a better option, but they are difficult, but there are some options coming available. So with that, I'd like to thank my lab, the Mito core at UMC, the histology core, the Department of Pharmacology and Toxicology, Women's Health Research Center, and the Mississippi CBRA for all their help. And I'll take any questions.